Hello. Very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible, and also let me know if the video is visible to you along with the PPT. Let me know if you can hear to my voice, if you can hear it properly, and also if you can see the PPT. Okay, the voice now, is it okay? Okay, fine. Thank you. I think there is little network issue. It will be fine. Meanwhile, let me know if there is any other issue or not. We will wait till it's 6 p.m. and then we will start today's session. Please wait till it's 6 p.m. and then we will start today's session. Okay, so it's almost 6 p.m. I think I will start with the session. Let me make the meeting open so that anybody can join. One more minute and then I will start it. Okay, so I think I will start today's session. So, very good evening to the students who have joined. My name is Mousumi Deb and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Organic Chemistry in Biology and Drug Development, and today it is our week 6 live session. So, in today's week 6 live session, what we will do is we will discuss the week 7 course content, like what we do in every class. So, we take the top week course contents which is live. So, right now, week 7 course content is live on the SWAYAM portal, and that is why I will today discuss week 7 course content in this our week 6 live session. Meanwhile, let me know if there is any problem for listening to my voice or seeing the PPT. If there is any problem with the audio or the PPT, please let me know in the chat box. Okay. Since I'm recording this session, I have muted you all and kept your cameras off so that there is no disturbance. But the chat box is always open. You can reach me through the chat box at any time. I'm checking the chat box only now and then. So if you have any doubt, any problem, or if you are facing any network issue, please let me know in the chat box. Okay. So before starting, let me point out few things which I do in before every class. So there will be a problem solving session every week, and these sessions will be on Google Meet platform. The code for the Google Meet class is the same which you are using for joining the today's class. The sessions will be on every Monday from 6 p.m. to 8 p.m. If any network disruption happens from my side, So please wait for few minutes. Do not leave the session. I will try to join as soon as possible. And if there is any network issue from your side, you can rejoin at any time. You don't have to worry about missing out anything since I'm recording the session. Okay. After the class, I will share this class recording and also the presentation which I am showing to you in the Google Meet. So the two things I will upload it and you can find it in the SWAM portal. I will show you where to find this class material, this presentation, video and audio at the and the PPT at the end of the session. Okay. So during the sessions, the students can post their question, their doubts through the chat box. Okay. So as I have told you that I am recording the session, that is why I have muted your cameras and microphones, but the chat box is always open, so you can reach me out through the chat box at any time. I will check your doubts, your queries, and even your answers, which you will giving for the questions I am showing. So everything we will communicate through the chat box. Okay. But if any doubt persists after class, you can drop an email to me. At the end of the session, I will share my email ID to you. This email ID, on this email ID, I will be available at any time. So if you have any doubt, any query regarding the class or any course content, you can let me know in that email. This presentation and class icon I have discussed that I will be sharing to you soon after the class. So today, as it is the week 6 live session where we will discuss the week 7. So in our first live session, we had completed week 1 and week 2 course content. And subsequently today, in our week 6 live session, we are going to look at the week 7 course content. So like every class, I will show you 20 MCQ question, and these questions will be based on the content which are in the week 7. Okay, and these content are right now live on the SWAM portal, and you have to submit the weekly assignment based on the week 7 course content soon, right? So this question which I am showing you today, it will help you to understand the week 7 course contents better, and it will also help you to answer the weekly assignment portions. Okay, so I hope everything is fine with my voice and also the presentation. If not, please let me know. Okay, I will try to adjust the things. Otherwise, I am going to start the session now. Okay. So first, let us see what are in the week 7 course contents. So the week 7 course contents are molecular biology and the chemistry of cofactors and coenzymes. So these two topics are we are going to discuss today. First is the molecular biology, followed by the chemistry of cofactors and coenzymes. Okay. So let us start with the first question of today's session. So the first question of today's session is, which of the following statements is false regarding the restriction enzymes? Your options are: number A, these are endonucleases; number B, these are exonucleases; number C, these only recognizes palindromic sequences; and number D, these digest the double-strand DNA at specific sites. So which of the following statement is false regarding the restriction enzymes? Again, I will repeat. Your options are: number A, these are endonucleases; number B, these are exonucleases; number C, these only recognizes palindromic syndrome sequences; and number D, these digest double-strand DNA or dsDNA at specific sites. So in between this four option A, B, C, and D, particular option is false regarding the restriction enzyme. You have to identify the false statement and let me know in the chat box A, B, C, or D which of the statement is false. I will give you 2 minutes and then I will check the chat box and I will show you the correct answer and we will discuss about the restriction enzymes in detail. Now let me check the chat box. Okay. See, even if your answer is wrong, it's fine, but answering is important because otherwise I won't understand that you are following the class or not. So whatever you think is the correct answer, please let me know in the chat box. Okay, it doesn't matter if your answer is wrong, but answering is little important. Okay, I will give you 2 more minutes. Let me know which of the following is the correct answer for problem number one. Which of the following statement is false regarding restriction enzyme? Okay, let me check the chat box one more time. Okay, I got a one answer which says it is number B. Let me check if number B is the correct answer or not. See, that's why answering is very important. Okay, thank you for answering. Now let me check if number B is the correct answer. Yes, number B is the correct answer. Very good. So these are exonucleases. These statements is false regarding restriction enzymes. Now let us understand what the restriction enzymes are. So what is a restriction endonuclease? So restriction endonucleases are family of enzymes that each cut the DNA at a specific sequence within the DNA, alternatively called restriction enzyme. So a restriction endonuclease or restriction enzyme can cut the DNA when it recognizes a specific sequence. These specific sequences are known as the restriction sites. So restriction, there are few points which you need to remember regarding the restriction enzymes. So these enzymes are endonucleases. Okay. So the statement number A is correct. These are endonucleases. These are not exonucleases. Okay. The recognition side is palindrome. So palindrome means a sequence which is same from both sides. So for example, Madam, I am Adam. If you see this word, Madam, I am Adam, so you will see that if it is spelled from forward or backward, it is same. So it is known as the palindrome. So the restriction enzyme, they recognizes palindromic sequences with only. So what does this palindromic sequence mean? Means that from 5 prime to 3 prime, the sequence is same for the both strand. Okay, so as you can see from here, the 5 prime A G C T 3 prime. And here also in the if you read it at the backward, the 5 prime A G C T 3 prime. So both strands are the same. So the restriction enzyme, they recognizes the sites which have the palindromic sequence. Okay. The recognition sites are different sites and unique sites. They cuts between same nucleotides on each strand. Okay. So what are the applications for restriction enzymes or restriction endonucleases? So the applications are like that. The isolated restriction enzymes are used to manipulate DNA for different scientific applications. They are used to assist the insertion of genes into the plasmid vectors during gene cloning and protein expression experiments. Restriction enzyme can also be used to distinguish gene alleles by specifically recognizing single base changes in DNA known as single nucleotide polymorphisms. Okay, so these are the applications of the restriction enzymes or restriction endonucleases. Okay, so statement number B, which has said that it is the exonucleases, this is false because restriction enzymes are endonucleases. These are also known as restriction endonucleases for that. Okay, so yeah, number B was the correct answer. Well, very good. So now I will move toward the next problem. So problem number 2 says only dash is a cofactor of the type 2 restriction enzyme. Fill in the blanks. You have to fill in the blanks in the statement which says only dash is the cofactor of the type 2 restriction enzyme. Your options are number A, copper 2 plus, number B, magnesium plus 2, number C, zinc plus 2, and number D, iron plus 2. So which of the following ions, copper plus 2, magnesium plus 2, zinc plus 2, iron plus 2 is the cofactor of type 2 restriction enzyme? You have to fill in the blanks. Again, I will give you some time. Think and then let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 2. What is the cofactor of the type 2 restriction enzyme? Copper plus 2, magnesium plus 2, zinc plus 2 or iron plus 2? Only dash is the cofactor of type 2 restriction enzyme. You have to fill in the blanks. I will give you some time. Then I will check the chat box. Again, even if your answer is wrong, it doesn't matter, but please answer which of the following you think is the correct one. So problem number 2, which says only dash is a cofactor of type 2 restriction enzyme. What will be the correct answer? Your options are number A, copper plus 2, number B, magnesium plus 2, number C, zinc plus 2, number D, iron plus 2. Let me check the chat box now. Okay, I've got one answer which says it's number B. Again, okay, I have two answers which says it's number B. Okay, let me check number B, is it the correct one or not? So the correct answer is very good. It's number B. Magnesium plus 2 is the cofactor. Only magnesium plus 2 is the cofactor for type 2 restriction enzyme. So what is the restriction enzyme? We have discussed in the last slide that these restriction endonucleases cuts the DNA at specific sites, right? So there are different type of restriction endonucleases. So what are this type? So we have type 1. These recognize specific sequences but then track along the DNA around 1000 to 5000 bases before cutting one of the strands and releasing a number of nucleotides around 75 where the cut is made. A second molecule of the endonuclease is required to cut the second strand of the DNA. For example, ECO-K. It requires magnesium plus 2, ATP and SAM. SAM means S-adenosyl methionine as cofactors for functioning. Type 2 restriction enzyme, they recognize a specific target sequence in DNA and then break the DNA both strands within or close to recognition site. For example, E. coli, this is usually required magnesium plus 2. And type 3, these are intermediate properties between type 1 and type 2. They break both the DNA strand at a defined distance from the recognition site. So for example, HGI. This requires magnesium plus 2 and ATP as the cofactor. So type 2 restriction enzymes are made up of a single subunit cleaved within at a short distance from DNA recognition sequence. They are simple enzymes having single polypeptide. They have separate methylase and endonuclease activity, requires mostly magnesium plus 2 as cofactor. This enzyme perform only the restriction function. It is the most common restriction enzyme used in gene cloning. The recognition and cutting site at the same one. They generally recognize 6 nucleotides, some recognize 4, 5 or 8 base pair also. For example, PVUI, PVUII, E. coli cleave the DNA to generate different ends. So these are the about the type 2 restriction enzyme. So again, we have a table regarding different type of in a tabular form of different type of restriction enzyme. Type 1, type 2, type 3 are most common, and we have a very less common type 4 restriction enzymes also, which are very less common. So nuclease activity, multimer and heterotrimer for type 1. The type 1, so these are the structure. Okay, the structure of type 1 is multimer or heterotrimer. The type 2 is homodimer. The type T is homodimer, and for type 4 it is not known. The recognitions side pattern: type 1 two sides in any orientation, type 2 small 4 to 8 base pair usually palindromic, and type 3 two sides in head to head orientation non palindromic. And the type 4 does not have any specific specificity, it has a weak specificity. Cleavage site: variable distance from recognition site, non-specific cleavage for type 1. For type 2, cleavage within the type 2p and outside type 2s, the recognition site. And type 3, cleavage of one strand, nicking activity of 24 to 25 base pair from recognition site. And the type 4 are methylated only. And cofactor: ATP magnesium plus 2 and SAM for the type 1, magnesium plus 2 for type 2, ATP magnesium plus 2 SAM for the type 3, and for type 4 we have ATP and GTP. Okay, so these are the about the different type of restriction enzyme. So now I will move toward the next problem. So problem number 3 says the palindrome sequence recognized by restriction enzyme E. coli is number A, AAGCTT, number B, CTGCAG, number C, GATATC, and number D, GAATTC. You have to identify the palindrome sequence which is recognized by the restriction enzyme E. coli. Your options are number A, AAGCTT, number B, CTGCAG, number C, GATATC, and number D, GAATTC. So which of the following palindrome sequence is recognized by E. coli? Let me know in the chat box, A, B, C or D, which of the following you think is the correct answer. The palindrome sequence recognized by restriction enzyme E. coli is, your options are number A, AAGCTT, number B, CTGCAG, number C, GATATC, and number D, GAATTC. Okay, let me check the chat box now. Okay, I have one answer which says it is number D. Okay, let me check if number D is the correct one. So the correct answer will be, yes, it's number D. Very good. So the palindrome sequence that is recognized by the restriction enzyme E. coli is GAATTC. So this table shows where the different restriction endonucleases or restriction enzymes cleave the DNA. The abbreviation R represents the purine. Okay, like adenine and guanine. The pyrimidines, cytosine, thymine, and uracil are abbreviated as Y, and the abbreviation W, it represents the adenine and thymine. Okay, so there are, as you can see in the last one, there are R, G, and Y, and and W also. So that is for that. So E. coli, the target sequence is 5 prime GAA TTC 3 prime, and it cleaves between the G and A. So as you can see here, in between G and A, it is cleaving. Ecore RV, it cleaves between A and T. Okay, as you can say, it cleaves between the T and A in the here in the same position. It cleaves. So it's 5 prime. The sequence, the palindrome sequence is 5 prime GA TA TC 3 prime. It is for Eco RV. HAE triple I, it is the 5 prime GGCC 3 prime, and it cuts between the G and C. Then we have hint 3, HIND triple I, so it cleaves between the A and A. Okay, the sequence it cleaves it 5 prime AAGCTT 3 prime. And we have PPUMI, which cleaves between the G and G, where G means your guanine. Okay, and C means cytosine. R means your purines. It can be any purine, and pyrimidines are abbreviated as Y, and W means adenine or thymine. So here, as you can see, it cuts between the G and G, means it cuts between the guanine. So this is the table which you need to remember for different restriction endonucleases. These are the target sequences and where it cleaves. Okay. So yeah, the correct answer was number D. Now I will move to the next problem. Problem number 4 says, which of the following restriction enzyme forms blunt ends? Your options are number A, E. coli, number B, HIND-III, number C, EcoRV, and number D, BEM-HI. So which of the following restriction enzymes forms the blunt ends? Your options are number A, EcoRI, number B, HIND-III, number C, EcoRV, and number D, BEM-HI. So which of the following restriction enzyme forms the blunt ends? I will be giving you 2 minutes, then I will check the chat box, and we will, after checking the chat box, I will show you the correct answer and we will discussing. Which of the following restriction enzyme from the blunt ends? Let me check the chat box. Okay, I have one answer which says it is number C. Okay, let me check if number C is the correct answer or not. So the correct answer is yes, it's number C. Very good. You are answering very well. So EcoRV, it is the restriction enzyme which form the blunt ends. So now let us first understand what are the blunt ends and what are the sticky ends or cohesive ends. So blunt ends are the ends when they cut both DNA strand at the same location. So when the particular restriction enzyme, they cut the DNA strand at the same location. Okay, the both DNA strand from one strand from 5 prime to 3 prime, another is from 3 prime to 5 prime. So when the restriction enzyme cut the both strand at the same position, we will have two completely normal or completely equal strands, or these ends. You can see these ends are blunt, and then So these are called the blunt ends. And sticky ends or cohesive ends, they produce staggered cuts in a single strand in sticky ends. Any DNA cut in the same enzyme will have ends with the same sequence. Means it will the sticky ends will form when the restriction enzyme cuts the two strands at the different position. So if I just show you the last slide, so here as you can see here it cuts this particular sequence is cut at 5 prime to 3 prime here, and on 3 prime to 5 prime it is cut here. So these ends are called the sticky ends. And eco RV, it cuts at the same location in the middle of the both strands, and as you can see it forms the ends which are known as the blunt ends. HAE triple I, it also forms blunt ends. Here HIND triple I, it forms the sticky ends. Okay. So restriction enzyme cuts each strand of two DNA stranded double helix at specific points in their sugar phosphate backbone, a little away from the center of the palindromic site, but between the two same bases in the both strand. As a result, single strand overhanging positions are left with two ends of DNA in the each strands are known as the sticky ends. Many restriction enzymes cut both strands of DNA at exactly same nucleotide position, especially in the center of the recognition sequence are known as the blunt ends. When they form hydrogen bonds with their complementary counterpart, they do not form hydrogen bonds. Stickiness and the ends facilitate the action of the enzyme ligase. For example, and no stickiness, only ligase enzyme helps to the joining. EcoRI is the example of sticky ends, and ALUI is the example of blunt ends. So this is the table. As you can see, EcoRV is the blunt cutter, AIUI is also a blunt cutter, SMA is a blunt cutter, EcoRI is a sticky end producer, and BAMHI is sticky end producer, RVPVUII is a blunt cutter, ASCI is a blunt cutter, DRAI is a blunt cutter. Okay, so these are the example of different blunt and sticky end producers of the restriction enzymes. Okay, so yeah, number C was the correct answer. Very good. Problem number 5 says reverse transcriptase requires which of the following for converting a single strand RNA into a double strand DNA. Your options are number A, all 4 NTPS, number B, all 4 DNTPS, number C, an RNA template, and number D, all 4 double DD NTPs. Reverse transcriptase requires which of the following for converting a single strand RNA into a double strand DNA? Your options are number A, all 4 NTPS, number B, all 4 DNTPS, number C, an RNA template, and number D, all 4 DDNTPS. Reverse transcriptase requires which of the following for converting a single strand RNA into double strand DNA? Reverse transcriptase requires which one of the following for converting a single strand RNA into a double strand DNA? Number A, all 4 NTPs, number B, all 4 DNTPs, number C, an RNA template, and number D, all 4 TDNTPs. Let me check the chat box. Okay, I have one answer. It says number C. Okay, let me check if number C is the correct one or not. So the correct answer will be, it's number B, all 4 DNTPs. Okay, now let me show you. So reverse transcriptase or ARTase is an RNA-dependent DNA polymerase, therefore produces a DNA strand from an RNA template. It has no associated exonuclease activity. So reverse transcriptase. So first we have a 5' to 3' single strand RNA template, or this RNA. Then we have the polymerization and DNTPase. What does this all 4 DNTPs, what will they do? They will form a RNA and DNA hybrid, okay, from 3' to 5', and then RNA is it will form a 3' to 5' single strand DNA from the RNA template, and then it will synthesize then again DNTPs, they will form a double from single strand DNA, it will form a double strand DNA. So that's how the reverse transcriptase works. Okay, so it requires the all 4 DNTPs form for converting a single strand RNA. Okay, a single strand RNA from a double strand DNA. Okay, because these DNTPs, these act as a units of the forming the DNA strand. So what is the difference between the transcription and the reverse transcription? So the sequences are, so your answer which you have given is that an RNA template. So the RNA template is already present. So from RNA template, the question was the converting single strand RNA into double strand DNA. So single strand RNA is the RNA template itself. Now I have asked that which for the following will be required for by this single strand RNA, which is the RNA template for synthesizing the double strand DNA. So the correct answer will be the DNTPs. All 4 DNTPs are required to form the double strand DNA. So now let me show you what is the difference between the transcription and reverse transcription. So transcription is the process of copying the genetic information stored in a DNA genome into a complementary strand of RNA, and reverse transcription refers to the reverse process of the normal transcription in which an RNA template is copied to form a cDNA molecule in retrophylosis. The encoding of DNA genome into nRNA is for the transcription, and for the reverse transcription, encoding of RNA genome into cDNA occurs in both prokaryotes and eukaryotes. The reverse transcription occurs in retroviruses. The transcription occurs in the cytoplasm of prokaryotes and nucleus of the eukaryotes. The reverse transcription occurs in the cytoplasm of the host. RNA polymerase is involved in transcription. Reverse transcriptase is involved. No primer is used by RNA polymerase in transcription. Lysyl tRNA acts as a primer for the reverse transcriptase. The produces transcription are used to protein synthesis. In transcription, products of the reverse transcription is integrated into the host genome. Okay, so these are the differences between the transcription process and the reverse transcription process. Now I will move to the next problem. So problem number 6 says identify correct statement. Number A, Tuck polymerase is used in PCR. Number B, any DNA polymerase can be used in the PCR technique. Number C, denaturation of DNA is the second step in PCR. And number D, none of these. So we have basically 3 statements. Number A, Tuck polymerase used in PCR. Number B, any DNA polymerase can be used in PCR. Number C, denaturation of DNA is the second step in PCR. And number D, none of these. So which of the statements is correct regarding the PCR technique? I will give you some time. Think and then let me know A, B, C or D, which of the following statement is correct regarding PCR. Is it A, B or C, or is D is the correct answer, which says none of these? Let me check the chat box now. Okay, I have not received any answer for this question. Okay, let me check which of the following is the correct answer. So, the correct answer will be number A. Tuck polymerase is used in PCR technique. This statement is correct regarding the PCR. So now let me show you the steps in the PCR. So the step 1 is the denaturation, which happens in the 95 degrees centigrade. Okay, the temperature for denaturation is 95 degrees centigrade. So what happens here? The double strand to DNA they are denatured. They are denatured and they are forming the single strand DNA. So two DNA strands are formed from the double strand DNA helix from the denaturation step. Then we have the DNA primers, and this DNA primers, the second step is the annealing, where the DNA polymerase, Tuck polymerase will be used. Okay, and nucleotides like DTTP, DCTP, DATP, and DGTP, different DNTPs, they will add up from 5 prime to 3 prime. Then Tuck polymerase, they will add we have first the primers. The primers will join at the 5 prime position, and then followed by the addition of different DNTPs and the repeat of the cycle, which will for 20 to 40 times to form new strands. So this is the total the steps. First step is denaturation, which happens in the 95 degrees centigrade. Second step is the annealing, which will happen in 55 degrees centigrade, and the step 3 is the synthesizing, which will happen in the 72 degrees centigrade. Okay, so Tuck polymerase. So why Tuck polymerase is thermostable? So these Tuck polymerase are used because these are thermostable. They can operate at a higher temperature. Generally, proteins, enzymes. So they do not operate in high temperature very much, but there are several enzymes which are thermostable. And in the Tuck polymerase are, for example, so these are commercially available thermostable DNA polymerases used in PCR to synthesize DNA. During PCR, the reactants are heated to 95 degrees centigrade. Normal DNA polymerase would be denaturated by at this high temperature. Tuck polymerases are derived from the eubacterium called Thermus aquaticus. The natural habitat of Thermus aquaticus is hot thermal springs with ambient temperature of 70 to 75 degrees centigrade. As Thermus aquaticus is adapted to live in extreme temperatures, the Tuck polymerase that is isolated from the bacterium is also capable of withstand high temperatures. Tuck polymerase can withstand high temperatures at the denaturation step of the PCR. Okay. So statement number A was the correct one. Now I will go to the next problem. Problem number 7 says which of the following statement is false regarding the PCR primers. Your options are number A, DNA polymerase requires primers to initiate the replication. Number B, primers are short and single stranded. Number C, primers are sequence specific, bind to particular genome sequence. And number D, higher the length of the primer, lesser will be the selectivity. Which of the following statement is false regarding the PCR primers? Again, I will repeat the options. Number A, DNA polymerase requires primers to initiate the replication. Number B, PCR primers are short and single-stranded. Number C, primers are sequence-specific and bind to particular genome sequence. Number D, higher the length of the primers, lesser will be the selectivity. So, which of the following statement is false? Again, I will wait for 2 to 3 minutes and then I will check the chat box. A, B, C or D, which of the following statement do you think is false regarding PCR primers? For which of the following you think is the correct option? Let me know in the chat box. Again, I will repeat which of the following statement is false regarding PCR primers. Number A, DNA polymerase requires primers to initiate replication. Number B, PCR primers are short and single-stranded. Number C, primers are sequence-specific, bind to particular genome sequence. And number D, higher the length of the primers, lesser will be the selectivity. Let me check the chat box. Okay. Okay, let me show you what is the correct answer and then let us discuss it. Okay. So the correct answer for problem number 7 will be number D. Higher the length of the primers, lesser will be selectivity. This statement is false regarding the PCR primers. So the considerations about the primer, the characteristic, the specificity. Specific for the intended target sequence, avoid the non-specific hybridization. The thoughts on the primer design is the uniqueness, length, base composition, and internal stability. Stability, form the stable duplex with the template under PCR condition. Melting the thoughts on primer design are melting temperature, annealing temperature, and internal structure. Compatibility, primers used as a pair will work under the same PCR condition, the primer pair matching. General rules for the primer design, primer and amplicon length. Primer length of determines the specificity and significantly affect its annealing to the template. Too short, too low specificity resulting non-specific amplification. Too long, decrease the template binding efficiency at the normal annealing temperature due to the high probability of forming secondary structure as hairpins. Optimal primer length should be 18 to 24 base pair general applications and 30 to 35 base pair for multiplex PCR. Optimal amplicon size 1300 to 1000 base pair for general application. Avoid less than 3 kb and 50 to 150 base pair in real time PCR. Avoid greater than 400 BP. Okay. These are the general rules for primer and primer design. Now I will move to the next problem. Problem number 8 says, which of the following statement is false regarding the plasmid PBR322? Your options are number A, PBR322 plasmid contains 2 genes. Number B, it is a natural plasmid. Number C, one gene confers resistance to ampicillin. And number D, other gene confers resistance to tetracycline. Which of the following statement is false regarding the PBR322 plasmid? Number A, PBR322 contains 2 genes. Number B, it is a natural plasmid. Number C, one gene confers resistance to ampicillin. And number D, other gene confers resistance to tetracycline. Which of the following statement is false regarding the plasmid PBR322? I will give you 2 minutes and then I will check the chat box, and after checking it, I will show you the correct answer. Let me check the chat box. Okay. Okay. Let me show you what is the answer for problem number 8 and then let us discuss about it. So the correct answer will be the statement which is false regarding plasmid PBr322 is number B. It is a natural plasmid. This statement is false. So PBr322 is a purpose-built plasmid vector and was one of the first widely used cloning vector. It has relatively small size of 4,363 base pair. That is important because transformation efficiency is inversely proportional to the size and above 10 kbp is very low. The nomenclature of PBr322, it is created in 1977 in the laboratory of Herbert Breuer and University of California, San Francisco. The P indicates the plasmid. BR, it indicates the Bolivar and Rodriguez, the two researchers who developed it. 322 distinguishes those plasmids from others like PBR 325, 327 developed in the same laboratory. The 3 PBR 322 is a 4,361 base pairs in length and has two antibiotic resistant genes. One gene BLA encoding the ampicillin resistant protein and another gene TA encoding the tetracycline resistant protein. It contains the originally replication of PMB1 and the ROP gene which encodes the restrictor of the plasmid copy number. The plasmid has unique restriction sites for more than 40 restriction enzymes. 11 of these 40 sites lie within the TAVT gene. These are two sites of restriction enzyme, HIND, III, and CLAI within the promoter of the TR gene, and there are six key restriction sites inside the AMPR genes. Okay. So remember, it is isolated from E. coli. As you can see, there are different restriction enzymes: E. coli, HND, I. Then we have one tetracycline resistant gene TETR, and one ampicillin resistant gene which is BLA. Okay, and this P indicates the plasmid. BR identifies there are two researchers who develop it, and C2 is a unique number. Now I will go to the next problem. So problem number 9 says which of the following enzymes is involved in the formation of recombinant DNA? Your options are number A helicase, number B DNA polymerase, number C ligase, and number D primase. Which of the following enzyme is involved in the formation of recombinant DNA? Helicase, DNA polymerase, ligase, or primase? Which of the following enzymes is involved in formation of recombinant DNA? Helicase, DNA polymerase, ligase, or primase? Let me check the chat box now. Okay, I have one answer which says it's number C. Let me check if number C is the correct one or not. So the enzyme which is involved in the formation of recombinant DNA is very good. It is ligase, number C. So what is the recombinant DNA and gene cloning? So recombinant DNA or rDNA is a form of artificial DNA that is created by combining two or more sequences that would not normally occur together through the process of gene splicing. Recombinant DNA technology is the technology which allows DNA to be produced by artificial means. The procedure has been used to change the DNA in the living organism and may have even more practical uses in the future. So we have first the donor DNA for restriction endonuclease. It is desired which takes out the desired DNA piece, and we have a plasmid DNA where restriction the plasmid DNA and the ligase they combines and forms the recombinant DNA. So protein encoded gene here we have and induced to the host cell multiplication of the several clone which will form. Another, I have a demonstration which says isolate the plasmid DNA and human DNA, okay, from bacterial cell, and then cut both the DNA stemples in the same restriction enzymes to form the sticky ends, and then mix the DNAs to join by base pairing the products of the recombinant plasmids of the many non-recombinant plasmids, and introduce DNA into the bacterial cell that have mutation in their own LAC-Z gene and place the bacteria in the agar containing ampicillin and X gel, incubate until the colonies grow. That's how the particular recombinant gene cloning happens. Okay. Yes, the number C, ligase, is the correct answer. These enzymes form the recombinant DNA from the desired DNA piece of the host DNA and the plasmid DNA. Now I will go to the next problem. Problem number 10 says, suppose you are doing a PCR to amplify the portion as indicated in the DNA. How many copies of the amplified portion of the double strand DNA will be obtained after 10 cycle? After 10 cycle, how many amplified portion of double strand DNA will be obtained? The options are number A 2 to the power 10, number B 2 to the power 8, number C 2 to the power 6, and number D 2 to the power 9. Let me check the chat box now. Okay. Okay. Let me show you the correct answer. The correct answer will be 2 to the power 8. So basically, you have to remember whenever this type of cycle is given, the formula will be 2 to the power n minus 2. Okay, so the 2 to here, the cycle is 10, means n is the 10. Okay, so the correct answer will be 2 to the power n minus 2, means 2 to the power 10 minus 2 is 2 to the power 8. So now RT-PCR stands for reverse transcription polymerase chain reaction, is a technique used in genetic studies that allows the detection and quantification of mRNA. Sensitive method that shows whether or not the specific gene is being expressed in given sample. RT-PCR is often confused with qPCR by students and scientists alike. However, the separate and distinct techniques. RT-PCR is used to quantitatively detect the gene expression through the creation of complementary DNA transcripts from RNA. qPCR is used to quantitatively measure the amplification of the DNA using the fluorescent probes. The principle in RT-PCR, the RNA template is first converted into the complementary DNA or cDNA using the reverse transcriptase. The cDNA is used as a template for the exponential amplification using PCR. Okay, one step RT-PCR versus two-step RT-PCR. In one-step approach, the entire reaction occurs in a single tube, and in the two-step reaction, the reverse transcription reaction, the um, PCR amplification can be performed in separate tubes. The difference between RT-PCR and traditional PCR is, although RT-PCR and traditional PCR both produce multiple copies of particular DNA isolates through the amplification and applications of the two techniques are fundamentally different. The traditional PCR is used to exponentially amplify target DNA sequences. RT-PCR is used to reverse transcript mRNA into cDNA and then amplify the result using the traditional PCR. There are several reasons why researchers would use this opposed to PCR. Since mRNA is the message that is sent to translation, RT-PCR can give us the measurement of the gene expression that PCR cannot. The cDNA produced does not contain any introns that DNA does. However, therefore, RT-PCR can provide us the genes that may be inserted into the prokaryotes, which cannot and do not splice into the introns out. This technique is also used to diagnosing this genetic disease as well as studying certain viruses whose genetic information is exclusively composed of RNA. Now I will go to the next problem. So problem number 11 says, which of the following statement is false regarding eukaryotic mRNA? Your options are: number A, they are derived from large RNA procedures. Number B, they result from extensive processing of their primary transcripts before serving as the translation components. Number C, they usually have poly A tails in their 3' end. And number D, they have both introns and exons in the matured form. Which of the following statement is false regarding eukaryotic mRNA? Again, I will repeat. Number A, they are derived from larger RNA precursor. Number B, they result from extensive processing of their primary transcripts before serving as the translation components. Number C, they usually have poly A tails in their 3' ends. And number D, they have both introns and exons in the mature form. Let me know in the chat box, ABC, which of the following you think is the correct one. Which of the following statements is false regarding eukaryotic mRNA? Your options are number A, they are derived from larger RNA precursors. Number B, they result from extensive processing of the primary transcripts before serving as translation components. Number C, they usually have poly A tails in the 3' end. And number D, they have both introns and exons in matured form. Let me check the chat box. Okay, I have one answer which says it's number D. Okay, let me check if number D is the correct answer or not. So the correct answer will be yes, it's number D. This statement which says that they have both introns and exons in the matured form is the false one. So RNA polymerase II transcripts long precursor RNAs. The pre-mRNA must be spliced into the mature mRNAs. This is a gene in DNA. We have exon, introns. So in pre-mRNA, after the transcription, we will have the exon and introns parts. But after splicing, we will have the spliced mRNA, which is the eukaryotic or matured, which only have the exons, not the introns. So what are the differences between the eukaryotic mRNA versus prokaryotic mRNA? The prokaryotic mRNA, the mRNA produced by the transcription of operons in the prokaryotes. Eukaryotic mRNA, the mRNA produced by the transcription of the single genes in eukaryotes. The synthesis of the mRNA occurs in the cytoplasm. The synthesis of the mRNA occurs inside the nucleus for the eukaryotic mRNA. The prokaryotic mRNA is the polycentronic and the eukaryotic mRNA is the monocistronic. The eukaryotic mRNA does not contain both 5' cap and 3' tail. The eukaryotic ones contain both 5' cap and 3' tail. The transcription is coupled with the translation in prokaryotes. The transcription of the eukaryotes occurs after completion of the transcription. This contains ribosome binding site, which are the Shine-Dalgarno sequence, the 5' cap of eukaryotic mRNA responsible from recruiting ribosomes. Lifespan of the prokaryotic mRNA is very short. The lifespan is relatively stable for the eukaryotic mRNA. The prokaryotic mRNA undergoes very little post-translational modification, but the eukaryotic mRNA undergoes major post-translational modifications. Okay. Now I will go to the next problem. Problem number 12 says, which of the following, consider the following diagram and identify the correct statement. Your option, this diagram you have to consider to identify the correct statement. The statements are number A, B is the template strand. Number B, A is the primer. Number C is the non-coding strand. And number D, A is the anti-sense strand. Which of the following statement is correct, which derived from this diagram? Let me check the chat box now. Okay. Okay, let me show you what is the correct answer and then let me. So the correct answer is A is the antisense strand. This is the correct statement. Okay, the A strand is the antisense strand. So as you can see, this is the DNA. Okay, from DNA we have two strands. One goes from the 5 prime to 3 prime and another goes from the 3 prime to 5 prime, right? So the strand which goes from the 5 prime to 3 prime is the antisense strand. Also it is known as the template strand. Okay, and the strand which goes from the 3 prime to 5 prime is the sense strand or non-template strand. This also I have discussed in the last class. Okay, so this strand A which goes from 5 prime to 3 prime is the antisense strand, and the strand which goes from 3 prime to 5 prime, the strand B, is the sense strand. Okay, and this is the RNA polymerase which synthesizes the mRNA transcript from the antisense strand from the antisense strand. Okay, so statement number D was correct. Now I will go to the next problem. Problem number 13 says, what is hollow enzyme? Your options are number A, inactive protein portion of the enzyme, number B, active complete enzyme, number C, non-protein portion of the enzyme, and number D, enzyme without any metal ion. Hollow enzyme, which of the following is the correct statement? What is hollow enzyme? Number A, inactive protein portion of the enzyme, number B, active complete enzyme, number C, non-protein portion of the enzyme, number D, enzyme without any metal ion. Which of the following is the hollow enzyme? What is the hollow enzyme? Your options are number A, inactive protein portion of an enzyme, number B, active complete enzyme, number C, non-protein portion, and number D, enzyme without any metal ion. Let me check the chat box. Okay, let me check what is the correct answer and then let us discuss about it. So hollow enzyme, correct answer will be number B. Active complete enzyme is known as the hollow enzyme. So first let us understand what are the coenzymes and cofactor. So a large number of enzymes require an additional non-protein component to carry out their catalytic functions are known as cofactors. A coenzyme or metal ion that is covalently bound to the enzyme protein is called the prosthetic group. Cofactors are of two types. Number one is the inorganic ions like iron, magnesium, manganese, zinc, and a complex organic molecule known as coenzyme. Some enzymes require both coenzymes and more metal ions for their activity. Coenzyme functions as transient carriers of the specific functional groups. So this is the apoenzyme, which is the protein portion. Now, what is a holoenzyme? So holoenzyme is basically the combination of apoenzymes and cofactor. Apoenzyme is the protein portion which is inactive. Cofactor is the non-protein portion which acts as an activator. And then we have the holoenzyme, which is the combination of apoenzyme, that is the protein portion, with the non-protein portion, that is the cofactor. And this holoenzyme is the complete enzyme, and it is a very active. Okay, and the main two terms are coenzymes and cofactor, where most of the students get confused. So now let us understand what are the differences of the coenzymes and cofactor. So coenzyme is a small organic non-protein molecule that carry common chemical groups between the enzymes. Okay, and cofactor is a non-protein chemical component that tightly or loosely binds with the enzyme and other protein molecules. Coenzyme is type of cofactor. Coenzymes and groups are two types of cofactor. Coenzymes are molecule, cofactors are chemical compound. Coenzymes are co-organic molecule, cofactors are inorganic compounds. Coenzymes loosely bound to enzymes, cofactors covalently bound to an enzyme. Coenzymes assist the biological transformations. Cofactor aid the function of the relative enzyme. Coenzymes serve as carriers to the enzyme. Cofactors, they increase the rate of the reaction of the catalyzed by the enzyme. Coenzymes can be removed from the enzyme easily. Cofactor can only be removed by the denaturing of the enzyme. For example, vitamins, biotin, coenzyme A are the example of coenzymes, and the cofactor examples include metal ions like zinc, potassium, magnesium. Okay, so these are the differences between the coenzymes and cofactor. Now I will go to the next problem. Problem number 14 says oxidation of retinol to retinal is catalyzed by number A, retinal dehydrogenase, number B, retinal reductase, number C, retinal isomerase, and number D, retinal deoxygenase. Oxidation to retinal is catalyzed by which enzyme? Number A, retinal dehydrogenase, number B, retinal reductase, number C, retinal isomerase, and number D, retinal deoxygenase. Let me check the chat box now. Okay. Okay, fine. Let me show you what is the correct answer for problem number 14. So the correct answer is the oxidation of retinol to retinal is catalyzed by number A, retinal dehydrogenase. So this is the all trans retinal ester. This is the structure of all trans retinal ester. So the first step is the ester hydrolysis. Okay, the ester hydrolysis forms the all trans retinol, which is the vitamin A. Then retinol dehydrogenase, it converts the all trans retinol to all trans retinal. Okay, and also there is a visual cycle reactions which form the all trans retinol to 11 cis retinal, which is for important for vision. And for H nu, this all 11 cis retinal is converted to all trans retinal, which forms to all trans retinolic acid from transcriptional modification and from aldehyde dehydrogenase, and also all trans retinol is converted to retinoid luciferin. Okay, so the conversion of retinol to retinal is catalyzed by retinol dehydrogenase. Okay, okay, it will be the retinol dehydrogenase. Okay, we have to. Okay, okay. Now I will go to the next problem. Problem number 15 says, which of the following statement is false regarding cystic fibrosis? Your options are number A, it is a genetic disease. Number B, PCR can detect the disease. Number C, in non-CF individual, CTFR gene codes for the protein that is sodium ion channel, and in the CF individual, means cystic fibrosis individual, mutation in CTFR gene leads to thick mucus secretion in the lungs. Which of the following statement is false regarding cystic fibrosis? Number A, it is a genetic disease. Number B, PCR can detect the disease. Number C, non-CF individual, CTFR gene codes for the protein that is sodium ion channel. And number D, in CF individual, mutation in the CTFR gene leads to the thick mucus secretion in the lungs. Which of the following statements is false regarding cystic fibrosis? Okay, let me check the chat box now. Okay, I have one answer which says it is number D. Let me check if number D is the correct answer for problem number 15. So the statement which is false regarding the cystic fibrosis is, it is number C. Non-CF individual CTFR gene codes for the protein that is in sodium ion channel. This statement is false. So gene mutations in cystic fibrosis. So for normal DNA, the CFTR protein develops normally, reach the cell surface and becomes open for the chloride ions. Okay. So it codes for the it generate the statement which says that there is sodium ion channel. It is actually for chloride ion channel. So this is the cell wall, this is the cilia, and this is the CFTR protein, which this is the healthy lung cell. And the chloride, in the case of healthy lung cells, what happens? The CTFR protein develops normally and reaches to the cell surface. Okay, it reaches to the cell surface and it becomes the open channel for a chloride. So ion. So what happens in the healthy lung cells? The chloride ion can pass out through the this CTFR protein channel and it can move freely. Cilia move back and forth, sweeping the mucus out of the lungs. The cilia it sweeps back and forth to sweeping the mucus out of lungs. When the gene mutation happens, door jamming mutation, including the G551D, the mutation affecting LoRa and Kate cleavage disables the function of the cell surface. The chloride ions can reach to the channel but the cell surface but cannot pass through because it has been called the doorman problem. CF lung cell with problem 1 for function, and then CF lung cell with two problems, location and function. With what happens, it's common delta F508 mutation. The CFTR proteins is made but it just floats in the cell without reaching to the surface. Okay, this mutant CF protein needs the corrected drug to boost the surface. It also dormant drug for the combat the open the channel so that the chloride ions can pass through. And because since the chloride ion cannot move freely and there is no operation of this this CFTR protein, what happens? The stick and the mucus buildup happens, which flattens the cilia. Okay, so this is the gene mutations which are the responsible for the cystic fibrosis. Okay, so remember, cystic fibrosis is a genetic disease which can be detected by PCR. In normal healthy lung cell, the CFTR proteins are in the surface, which opens for the chloride ions. But in the mutated ones, when there is a door jamming mutation, the CFTR reaches to the surface but does not open. And in case of the common delta F50 mutation, the CF protein, it floats inside the cell, it does not reach to the surface. Okay, so now I will go to the next problem. So problem number 16, it says that conversion of all transretinal to 11 cisretinal happens in options are number A blood, number B retina, number C pupil, number D none of the above. So where the conversion of all transretinal to 11 cisretinal happens? In blood, in retina, in pupil, and none of the above. Conversion of all trans retinal to 11-cis retinal happens in which portion? Let me check the chat box. Okay, let me show the correct answer. So the answer will be number B, retina. So this is the world's visual cycle where we have the rhodopsin, which is the 11-cis retinal opsin, which is formed in presence of light. It forms the Barthorp's rhodopsin, then lumirodopsin, then metarodopsin, then metarodopsin 1. Then it forms the opsin, and opsin goes to the 11-cis retinal opsin again in the world's visual cycle. This metarodopsin is forms all transretinal, which goes to the retinal retinal all cis retinal by the enzyme retinal isomerase in retina. Okay, and this all trans retinal is also formed from the diet to liver to all cis trans retinol. Then we have retinol dehydrogenase, which forms the all trans retinol from retinal from all trans retinol, and the NADP plus is formed convert to NADPH plus H plus. So this is the reaction, or this is the all trans retinal. So as you can see, there are all the double bonds are in the trans position to each other. Okay, and then now we have the water, and it cleaves this position, it attacks this position. It is the SN2 kind of attack, and it forms the pro cis retinal. Okay, and this is the reaction, how the all trans retinal is converted to all cis retinal. So remember, the enzyme is the all 11 the retinal isomerase, and the position where it happens is the retina. Okay, now I will go to the next problem. Problem number 7 says, which of the following is the coenzyme form of vitamin plantothenic acid? Your options are number A, pyridoxal phosphate, number B, tetrahydrofolate, number C, thiamine phosphate, and number D, coenzyme A. Which of the following is the coenzyme form of vitamin plantothenic acid? Your options are number A, pyridoxal phosphate, number B, tetrahydrofolate, number C, thiamine phosphate, and number D, coenzyme A. Which of the following is the coenzyme form of vitamin pantothenic acid? Pyridoxal phosphate, tetrahydrofolate, thiamine phosphate, coenzyme A. Let me check the chat box. Okay, I have one answer which says it is number C. Let me check if number C is the correct answer or not. So the correct answer is, it's number D. Coenzyme A is the coenzyme form of vitamin plantothenic acid. So this is a chart of the different vitamins with coenzyme and group transfer reactions. So B1 is the thiamine, coenzyme is the thiamine pyrophosphate or TPP, and the group transfer, it is the C2 aldehyde and decarboxylation. B2 is the riboflavin, the coenzyme is flavin adenine mononucleotide or FNN, and flavin adenine dinucleotide FAD. The group transfer is the hydrogen. Then we have B3 niacin, which is the nicotinamide adenine dinucleotide NAD+. The group transfer is hydrogen. Then nicotinamide adenine dinucleotide phosphate or NADP+. Then the group transfer is hydrogen. Vitamin B5 plantothenic acid, coenzyme is coenzyme A, and the group transfer is acyl. B6 is the pyridoxine. The coenzyme is pyridoxal phosphate. The group transfer is amino and decarboxylation. B7 is biotin. Biocytin is the coenzyme. Carboxyl is the group which is transferred. Vitamin B9, folic acid, tetrahydrofolic acid is the coenzyme and formyl is the group which is transferred. B12, the cyanocobalamin, B12 is the coenzyme and B12 coenzyme is the coenzyme and carboxyl HX rearrangements. There is a group transfer reaction which it catalyzes. So this is the plantothanic acid which converts the reaction is a plantothanic acid kinase enzyme which converts plantothanic acid to 4' phosphoplantothanonic acid. Then we have the plantothanial cysteine synthetase, which convert this it to the 4-phosphoplantothanonial cysteine. Then we have 4-phosphoplantothanonial cysteine decarboxylase, which it converts it to 4'-phosphothane. Then we have ATP plantothane 4-phosphate adenotransferase, which converts the 4'-phosphoplantane to 4'-D-phospho coenzyme A. Then D-phospho-coenzyme A is converted to coenzyme A by the D-phospho-CoA kinase, and coenzyme A is converted to hollow acyl carrier protein with phosphomethane as an active group by this CoA apo-acyl carrier protein plantothanial phosphate transferase. Okay, so this is the how the plantothanic acid is converted to CoA and the hollow enzyme. Now I will move toward the next problem. Problem number 18 says which of the two reactions are required to biosynthesize the first derivative of vitamin D inside the body. Options are number A, disrotatory ring opening followed by 1,5 hydrogen shift. Number B, conrotatory ring closing followed by 1,5 hydrogen shift. Number C, disrotatory ring closing followed by 1,7 hydrogen shift. And number D, conrotatory ring opening followed by 1,7 hydrogen shift. Which of the following two reactions are required to biosynthesize the first derivative of vitamin D inside the body? Options are number A, disrotatory ring opening followed by 1,5 hydrogen shift. Number B, conrotatory ring opening followed by 1,5 hydrogen shift. Number C, disrotatory ring opening followed by 1,7 hydrogen shift. And number D, conrotatory ring opening followed by 1,7 hydrogen shift. Which of the following two reactions are required to biosynthesize first derivative of vitamin D inside the body? Let me check the chat box. I have one answer which says it is number D. Let me check if number D is the correct answer or not. So the correct answer is very good. It is number D, conrotatory ring opening followed by 1,7 hydrogen shift. So this is the 6-pi electrocyclic ring opening and closure for thermal and photochemical condition. So first, as you can see in the photochemical condition, this is the 6-pi electrocyclic ring closure. We have 6-pi electrons here, 2 here, 2 and here 2. Total 6-pi electron we have. So whenever there are 6-pi electron, it is known as 6-pi electronic ring closure and ring opening. So in the photochemical condition or HNU condition, it is the conrotatory movement, and in the thermal condition it is the disrotatory movement. Conrotatory means the movement of the bonds will be, or movement of the particular orbitals will be in the same direction, and disrotatory means both the orbitals will move to the opposite direction. Okay, and this is the example of how the hydrogen shift happened. So it is known as a 1,5 hydrogen shift. Why 1,5 hydrogen shift? So in case of hydrogen shifts, the bonds which is broken is number as 1 and 1. then we have 2, then 3, then 4, and then 5. So as you can see, the hydrogen shift is happening from 1 position to 5 position. That is why it is known as the 1,5 hydrogen shift. And here, if you number it as it is, so you will see it is the 1 to 7 hydrogen shift. 4, 5, it is 6, and it is 7. So that is why it is known as the 1,7 hydrogen shift. So now in case of the vitamin D biosynthesis, as you can see, this is the 7-dehydrocholesterol, which in presence of UV or sunlight, it follows the 4-pi conrotatory movement. And then we have the vitamin D3, and then followed by 1,7 hydrogen shift, which converts this pre-vitamin to cholecalciferol vitamin D3. Okay. So the correct answer was number D, conrotatory ring opening followed by 1,7 hydrogen shift happens in the as a first step of first derivative of vitamin D inside the body. Now I will go to the next problem. So problem number 19 says the biosynthesis of gamma aminobutyric acid or GABA from glutamic acid involves number A, glutamate carboxylate as enzyme and biotin as cofactor. Number B, glutamate decarboxylase as enzyme and TPP as cofactor. Number C, glutamate decarboxylase as enzyme and PLP as cofactor. And number D, glutamate carboxylase as enzyme and PLP as cofactor. Which of the following is the correct statement regarding the biosynthesis of gamma aminobutyric acid or GABA from glutamic acid? Number A, glutamate decarboxylase. Glutamate carboxylase as enzyme and biotin as cofactor. Number B, glutamate decarboxylase as enzyme and TPP as cofactor. Number C, glutamate decarboxylase as enzyme and PLP as cofactor. And number D, glutamate carboxylase as enzyme and PLP as cofactor. The biosynthesis of gamma aminobutyric acid from glutamic acid involves which enzyme and which cofactor? Let me check the chat box. Okay, I have one answer which says it's number C. Let me check if number C is the correct answer or not. The correct answer is yes, it is number C, glutamate decarboxylase as enzyme and PLP as cofactor. So, gamma aminobutyric acid acts as an inhibitory synapse in the brain by binding to the specific transmembrane receptor in the plasma membrane of the both pre and postsynaptic neural process. It is the most highly concentrated in the substantia nigra and globus pallidus nuclei of the basal ganglia, followed by the hypothalamus and periaquedular gray matter. The gamma aminobutyric acid or GABA is the major inhibitory neurotransmitter in the mammalian central nervous system, present in the nerve terminal released from the electrically stimulated neuron, and it functions by binding to specific transmembrane present in the plasma membrane. So this is the overall reaction. Okay, so we have the glutamate, and the glutamate is then it's converted to the GABA for in presence of glutamate decarboxylase as an enzyme, and here we have the PLP as cofactor. This GABA is converted to the succinate semiannuate, and there are different reactions which is happening. So you don't need to memorize all the reactions here. Okay, so like for example, this GABA is converted to the succinate semialdehyde, which is then converted to succinate in the presence of succinic semialdehyde dehydrogenase, and so on. Then we have the GABA pyruvate transaminase, which converts the pyruvate to glutamate, and this is the cycle which it follows. Okay, so remember that glutamate decarboxylase is the enzyme which catalyzes the conversion of glutamate to gamma aminobutyrate, and here the one proton is converted to carbon dioxide. Here this is the decarboxylation type of reaction. Okay, now I will go to the last question. It says that in Huntington disease, the trinucleotide sequence that repeats more than 36 times is number A CAA, number B AGC, number C CAG, and number D TAC. Which of the following trinucleotide sequence repeats more than 36 times in Huntington disease? In Huntington disease, the trinucleotide sequence that repeats more than 36 times is CAA, AGC, CAG, and TAC. In Huntington disease, which trinucleotide sequence repeats more than 36 times? CAA, AGC, CAG, or TAC? Let me check the chat box. Okay. Okay, let me show you the correct answer and let us discuss about it a little bit. So the correct answer is CAG. This trinucleotide sequence repeats more than 36 times in Huntington disease. So first, what happens in case of Huntington disease to identify it? First, the blood sample is collected, which is tested with PCR to identify if there is a CAG's repeat more than 36 times. If there is a more than 30, then Sanger sequencing happen, which checks the heterozygous is SNP are there. Then extended CAG repeat and SNP is the same allele. This is eligible for allele selective therapy. So in normal CAG get less than 36 times is not in case of Huntington disease, but in case of Huntington disease, what happened? The extended CAG greater than 36 times it happens. Okay. So clinical features of Huntington disease is the motor, the cholera, the clinical features are the cholera, dysentery, motor impression, dysphagia, poor posture, incoordination, impaired gait and balance, delayed initiation of voluntary success. Cognitive is the loss of excessive functions, difficulty in multitasking, rigid and inflexible thoughts, poor concentration, easily distracted, impaired spatial perception, impulse control, disinhibition, perception of vocabulary, poor insight, short-term memory loss, and psychiatric is a depression, anchoria, decreased libido, suicidal institutions, social isolation, and so on. So these are the different clinical features of Huntington disease. So remember, in case of Huntington disease, CAG trinucleotide sequence repeats more than 36 times. Okay. So this was the last question of today's session. So today in our week 6 live session, what we did was we discussed about many things regarding the molecular biology and followed by the coenzymes, cofactor and all. So today it was the week 6 live session of today's session where we discussed about week 7 course content. Now I will share my email ID. So if you have any doubt or any query, you can let me know through the email regarding today's class. Okay. So this is my email ID. Okay, so there is a typographical error. I will just share it one more time. So this is in my email ID. So if you have any doubt, any query, you can reach me out through the email. And where to find this kind of problem solving session, I will show you once. So you have to first log in to the SWAYAM portal. Okay. So when you log into the SWAYAM portal, you will find there is a problem solving session here in the announcement section. And if you click on that, you will have the problem solving session recording. By clicking here, you will go to you will directed to an Excel sheet where you will find the class recording and also the presentation. So today now I will close the session for today. If you have any doubt, any query, feel free to reach me through the email. Thank you for joining, and we will meet on the next Monday from this time 6 p.m. to 8 p.m. And one more thing, please, this Gmail ID will be same for the next class also. So thank you. I will leave the session for today. And if you have any problem, please let me know in the chat in the email, or you can let me know through the discussion forum also. So thank you. I will close the session for today.